Hello, everyone, and welcome to today's AAPS eChalk Talk. I'm very excited to welcome you to our talk entitled Cell and Gene Therapy Optimization, a Framework Assessing Efficacy and Safety with Dynamic Biomarkers. This talk is sponsored by Sapient. Cell and gene therapies hold enormous potential as new treatment modalities for traditionally difficult to treat diseases. There are still challenges, however, stemming from heterogeneity in target engagement and drug response across patients, as well as adverse events. This presentation will explore use of discovery mass spec to identify dynamic biomarkers of response and adverse events throughout the course of cell and gene therapy. To kick things off, I'd love to introduce our speaker, Dr. Jain, founder and CEO of Sapient. Dr. Jain is a physician scientist with more than 20 years of expertise in physiology, computational biology, and mass spectrometry-based bioanalytics. He formed the Jain Laboratory at the University of California, San Diego, where he led his team to develop next-generation mass spec systems to probe the non-genetic landscape of disease across large-scale human studies. Dr. Jain founded Sapient in 2021 as a spin-out of the Jain Laboratory to expand upon his mission. I know I'm looking forward to hearing what he has to share today, so Mo, please take it away.
Thank you so much for the kind introduction. We are really excited to be here today to talk about this emerging field that's just exploded over the last several years. And that, of course, is cell and gene therapy. And to really provide a framework for how we think biomarkers may help accelerate both the development and the deployment of these emerging technologies and therapeutic modalities. So just to begin with, thank you all for joining us today. As you're all well aware, cell and gene therapy is an area that has absolutely exploded over the last several years. Surprisingly, there's over 1,500 registered clinical trials that are currently ongoing in this space that spans both cell therapy, gene therapy, DNA and RNA therapeutics, as well as gene-modified cell therapeutics and genome editing. As many of you who followed the field are quite familiar, there are a number of these now that are going before the FDA, certainly a greater number than ever have before. And it's been proposed that the FDA is likely to approve up to 9 of these cell and gene therapies in 2023 and certainly entering into the first part of 2024. So there's a tremendous amount of excitement around this new area and this new therapeutic modality, particularly, as you had mentioned, for difficult to treat diseases, rare conditions, and neoplastic disorders. Now, I think this quote from the FDA really summarizes the current state of affairs in cell and gene therapy. And just to read it out, we believe these cell-based and gene therapy technologies hold tremendous promise for addressing some of the most intractable diseases. But with their novelty also comes new uncertainties and some unique risks. And that's what we're here today to discuss, how we may be able to bridge some of these uncertainties and risks using biomarker-based discovery. Now, anyone who's followed the AAPS over the last year is quite familiar with a number of rather severe adverse events that have occurred in the setting of cell and gene therapies. And certainly these always damper some of the underlying promise. But we very much believe that with proper study, these therapeutics can become the standard of care for many, many intractable diseases for which we currently do not have treatment. Now, if we look at this holistically across a meta-analysis of the 250 plus clinical trials that are currently ongoing, for instance, in the gene therapy space that use recombinant AAV vectors, what we find is that over 30% of these trials had rather severe treatment emergent adverse events. And these span the therapeutic areas, whether it be ocular, CNS, liver, muscle, LSD, as well as other clinical trials. And you can see within each of these areas, approximately a third of the trials have had reports of serious adverse events. And it's really important that we think about these adverse events, we embrace them, we begin to understand how we can identify individuals at risk for these serious adverse events, and we removed those individuals, or at least we mitigate these events as quickly as possible as a modality for developing these therapeutics. In many ways, understanding these adverse events is as critical as developing the therapy itself. Now, when we look at gene therapies, there's a number of potential inciting etiologies which can have important impact on both the acute as well as the durable response to these therapeutic modalities. So when we think about pre-existing cases, whether they be NAbs, T-cell-based immunity, disease-associated immunity, and prior infections, these are obviously pre-existing conditions which can influence our response to gene-based therapies, as well as both acute innate as well as adaptive events that may occur post-exposure, both immediately as well as days to months later, which can also very much influence our response. Now, as many of you are well aware, both these pre-existing acute as well as more chronic states of inflammation and immunity can influence greatly our response as well as the durability. These can be rapid in the case of an innate immune response, which can result in acute toxicities, or much more gradual, causing vector dilution as well as long-term impairment of response to these therapeutics. Now, from an actual gene editing perspective, as you're well aware, there's also been quite a bit of discussion in the literature regarding off-target effects and ensuring proper silencing of the target gene while minimizing these off-target effects that may occur for secondary genes of interest. Similarly, in the cell therapy space, there's a number of challenges that absolutely must be considered. With autologous CAR-based therapies, for instance, there's a well-described cytokine release syndrome or immune effector cell-associated neurotoxicity syndromes, both of which relate to underlying autoimmune reactions that occur in the setting of cellular-based therapies. There's also concern for cells that are introduced into an individual with regards to trafficking and infiltration into tissues and tumors of interest, both of which can limit efficacy, particularly for solid tumor types. When we think about allogeneic CAR therapies, similarly, there's a cytokine release syndrome, as well as toxicity related to intense lymphodepletion. There have been cases of graft versus host disease, as would be expected, as well as rejection of the allogeneic cells. Again, all of which limit both the benefit that is observed in the setting of these therapeutics, as well as incite initial adverse events that can be quite toxic to the patient. Now, when we think about all these adverse events, whether they occur in the setting of gene therapy or cell-based therapies, these are really dynamic processes, as all drug responses really are dynamic. Under ideal circumstances, individuals at baseline who have clinically apparent disease are receiving an intervention, whether it be a cell or based gene-based therapy. Some portion of these individuals are going to have clinical improvement, which is wonderful. Some portion of these individuals are going to have minimal clinical improvement. And then, as mentioned, there's a subset of individuals that are going to have mild adverse events. Typically, these are not dose or treatment limiting. And then there's a subportion of individuals, as we had discussed previously, that occurs in approximately a third of trials to date, in which severe adverse events can occur. Typically, these can be quite limiting with regards to the therapeutic efficacy as well as dosing of these treatments. Now, there's a number of steps here where it's critical to be able to leverage biomarkers as part of the development process, both in identifying individuals who are having mild or severe adverse events at the earliest potential time points when secondary therapies may be instituted, as well as at baseline, being able to identify those individuals who are likely to have maximum clinical improvement and minimum adverse event as a way of expanding, if you will, the therapeutic index. Now, as you can imagine, responses to the same type of intervention, again, whether it be a cell therapy or a gene therapy, vary substantially across people and over time. And given the underlying dynamic nature of this process, it's critical that we have dynamic precision biomarkers that allow us to subset and identify individuals, as well as longitudinal sampling that allows us to identify these precision-based markers. And this is where small molecule biomarkers can be particularly advantageous in the setting of cell and gene therapy. These are thousands of small molecules that are floating around in our blood at all times. And these small molecules are particularly dynamic and tuned, if you will, to read out processes that are critical for cell and gene therapy. To start with, these small molecule biomarkers read out both the genetic and non-genetic factors that contribute to human disease as well as drug response, whether they be advantageous responses or negative adverse responses, including induction of the immune system. These markers are produced in the normal tissue, they're produced in the diseased tissues, they're produced in the microenvironment, and they're also introduced in cells that are introduced as part of cellular therapies. Now, given the physical chemical properties of these small molecules, while they're produced intracellularly, these are released from the intracellular state through the organic transporters into the blood. And by assaying these thousands of markers within the blood, we can have deep insight and glimpse into what's actually occurring, once again, in the normal tissue, in the diseased tissue, in the microenvironment, or in cellular therapies that have been introduced into people. These markers are highly dynamic, which is advantageous and allows us to read out in real time what's occurring with cell and gene therapies or changes in disease processes. And from a practical perspective, these are highly conserved across species. And so we have the ability to assay both humans, preclinical models, cellular models, as well as reverse translate in either direction, which becomes extremely advantageous when we think about translation of these biomarkers. Now, this is not a new idea of leveraging these markers for the sake of cell and gene therapies. The real limitation has always been about how do we assay them. And this is where Sapient has made a number of advances, particularly with our introduction of what we call high throughput mass spectrometry. These are rapid liquid chromatography, or RLC systems, coupled to high resolution QTOF ion mobility mass spectrometry, or RLC-MS. Here's a video of our laboratory. We're based here in San Diego, and you can see the stack of these instruments that allow us to take any biological specimen, whether it be a cell study in vitro, whether it be a preclinical model, whether it be human samples, both blood, urine, CSF, tissue, tumor, cells that have been introduced into individuals. And in any of these biological specimens, we can very rapidly assay 15 to 20,000 small molecule biomarkers. Now, these cross the diverse chemistries that are present in people, from very polar water-soluble molecules all the way through very neutral lipids. And so you can think of this as combined metabolomics and lipidomics in one. We have enormous capacity and high throughput on these systems, which allow us to handle thousands of samples per day, and a capacity of over 1 million samples per year on our single site here based in San Diego. Now, from these thousands of markers that we're assaying in biological specimens, over 700 of them we can structurally identify upfront using pure commercial standards. And for those tens of thousands of markers that remain quote unquote unknown, we're obtaining what's called dense 4D chemical characterization. This includes their retention time on column, their accurate mass MS1, and secondarily their chemical formula, their MS2 chemical fingerprint, and their CCS on ion mobility mass spec. Now, these 4D chemical characterization allows us to identify a unique molecule, to map it, to measure it under a CLIA-CAP laboratory, or to develop an FDA-approved assay for this singular molecule, as well as to isolate out that marker and begin to study it and structurally elucidate it as needed. These systems, as you can imagine, are incredibly robust and fast. And in order to ensure the robustness, they're all embedded in a very rigorous quality matrix that allows for real-time monitoring of over 500 different parameters, from pooled replicate blinded samples that are injected simultaneously to exogenous standards that are spiked into every sample that allow us to monitor both the production and the performance of our instrumentation, our column chromatography, our sample preparation and robotic handling, as well as the actual integrity of the sample itself. Now, while these tools were initially optimized and developed around the small molecule biomarkers that I just had discussed, and these also are now being leveraged in our laboratory here for simultaneous mass spec-based proteomics and high sensitivity multiplexed cytokine measurements. And I'd be happy to chat with anyone offline who may be interested in these additional modalities. Now, as you can imagine, these technologies generate a massive amount of data very, very quickly. And it's not the goal to generate data for the sake of data's sake, but rather to take this massive amount of data and to leverage it in a manner that allows us to derive key actionable insight, essentially going from data to true knowledge. And this is what allows us to advance forward a cell and gene therapy program. Now, this bridge between data and knowledge is really spanned by deep biocomputational analysis. And so we have expertise not only in the generation of this data, as I just discussed, but also in the analysis of this data that's done together with our clients and sponsors. Our team consists of a team of experts in data analysis that have deep experience in handling of complex mass spectral data and the integration of this with multidimensional data, including genomic measures, proteomic and cytokine measurements or inflammatory markers, longitudinal clinical outcomes, preclinical measurements, including in preclinical models and pharmacogenetic modeling, clinical laboratory values, human biological outcomes, and either omics data, including the microbiome, which may be critical for these types of therapies. Now, there's a variety of different questions and approaches we can take in the deep statistical analysis of complex data, everything from traditional regression and Bayesian statistics all the way up through much more advanced machine learning and AI-based tools that allow us to identify new targets and to validate existing targets, that allow us to identify diseases at their earliest time points, to understand adverse events, whether they be mild or severe, once again, at their earliest time points, to understand disease progression, to begin to stratify patients according to responders, non-responders at the earliest time points as a means for enriching clinical trials and improving outcomes as a whole, as well as for leveraging these tools for target engagement, particularly in the setting of gene therapy, as we'll talk about in just a moment. Now, to support these discovery efforts are some very unique data assets internally here at Sapient that have been amalgamated in what we call our human biology database. This is an expansive human data repository that's been amalgamated from over 100,000 human biological specimens that have been collected from around the world, which have already been analyzed on our mass spectrometry systems to measure these tens of thousands of markers. Now, this data has all been collected together with orthogonal genomics data, clinical outcomes data over 3 decades of time, protein and cytokine measurements, lifestyle factors, dietary factors, microbiome factors, as well as intense measures regarding response to various therapeutics. Now, this data set includes now over 150 million phenotypic data points and represents a very unique tool that we can take and leverage in order to amplify a discovery that may come from one of your data sets, in which we're making a discovery potential in a small subset of samples on the order of 100 to several hundred samples, and then taking that finding and asking how does it validate or behave now across more than 100,000 other individuals from around the world. And in this way, we can amplify and accelerate the underlying discovery process. Phenotypic data in this database includes over 70 various diseases and tissue-related injuries that you can imagine are quite advantageous for understanding adverse events, as I'll talk about in just a moment. Now, as I mentioned, we spend quite a bit of time thinking through and leveraging multidimensional data, and in particular, integrating complex data sets, including genomics and small molecule biomarkers. And this can be essential within the setting of both cell therapies, where we may be targeting particular tumor types and mutational burdens, as well as in gene therapies, where we may be selectively silencing a particular genetic variant. Now, within our database, we have over 20,000 people in which we have dense genotyping with corresponding blood samples that have already been analyzed on our mass spectrometry systems. Now, across these individuals, there are over 70,000 different small molecule circulating markers that have been assayed. And when we take this complex small molecule biomarker data and analyze it within the context of a human genome, what we find is really quite remarkable. For approximately half the markers seen in human blood, we can find a genetic variant that associates it with it at a genome-wide threshold and allows us to read out particular genetic variants of interest. This is extremely powerful for once we identify a marker, understanding its biosynthetic and tissues of origin. Correspondingly, we can go in the opposite direction. When we think about the genome as a starting point, if we look at the 20 million or so common nucleotide polymorphisms, over 5% of them associate with a circulating biomarker in blood. Now, this becomes even greater if we think about enrichment for those common polymorphisms that occur within the coding region or the transcribed region of a gene. These are the common polymorphisms that are thought to alter actual protein function. And when we look across the 20,000 or so genes within the human genome, we find that over 90% of these genes actually have a circulating biomarker in blood that associates with them, again, at a genome-wide threshold. Now, this is critical. If we're trying to silence a particular drug, we can identify a circulating marker that reads out the actual function and silencing of that drug within a particular tissue bed. And that becomes instrumental for understanding target engagement and efficacy of gene therapies, as well as in the cell therapy space. Now, I'm hoping we can just take a moment here and talk through a framework for how we begin to apply these small molecule biomarkers, specifically within the context of cell and gene therapies. And there's some nuances for each of these. So starting first within the gene therapy space, as you can imagine, there's a number of potential applications whereby these approaches may be instrumental. And I just wanted to highlight three of them here today. The first is in patient selection. As we know for many of these gene therapies, including in the rare disease space, these operate and work best in a subset of individuals. And being able to identify those individuals upfront using biomarkers, and then particularly engage them with the context of these therapies is really instrumental in being able to expand out our therapeutic index. And so we think a lot about patient selection and companion diagnostics. As I also mentioned, within the context of gene therapy, being able to leverage these biomarkers to understand target engagement and pharmacodynamic response is absolutely essential, in particular ensuring effective silencing or amplification of a gene of interest in a tissue of interest. And then finally, thinking through drug-induced toxicities I'd mentioned earlier, both identifying those individuals up front who may be predisposed to various drug-induced toxicities, including in the setting of AAV vector use, as well as identifying those individuals at later time points who may be prone to innate or adaptive immune activation in the setting of these gene therapies, once again, as a means for expanding out our therapeutic index. Now, here's one such example of how we can go about leveraging our genetic data sets for target engagement. This surrounds the PD-1 pathway, which has obviously been transformative in immunotherapy and activation of the immune system for solid tumors. And the way we went about trying to understand sort of these precision biomarkers, as you're well aware, a subfraction of individuals who receive these immunotherapies actually end up having a durable long-term response to these therapies. And so we've been very interested in trying to identify submarkers, particularly at baseline, that may denote individuals who are most likely to have a benefit in the setting of these IO therapies or to have a maximum target engagement. And now the way we began this project was through genetic interaction and looking at common genetic variants that occur in the PD-1 pathway, both in PD-1 and PD-1L. And when we look at genetic variants in these markers and look across the 70,000 or so biomarkers within our database, we find that approximately a handful of these, as shown here on the top modified Manhattan plot, are highly associated with genetic variants in the PD-1 pathway. Now, two of these markers that are circled there in blue are particularly interesting for chemical reasons, and we have reasons to believe that these are highly specific for the PD-1 pathway, once again, reading out a marker in blood for the actual activation state of this particular biological pathway. Now, when we look in our database before and after treatment, what we find is that the singular marker is increased after therapy, as you see on the bottom left image. So if we look before and after treatment, these markers are acutely upregulated in blood following activation and exposure to these drugs. And we can see that even among those individuals post-treatment that have activation of this pathway, there's quite a bit of heterogeneity, as we know clinically, in which there's some individuals that have a very robust activation of the PD-1 pathway, where there's others that have a much more subtle elevation. Now, if we substratify individuals according to this one biomarker that's, again, reading out the PD-1 pathway post-exposure to an immunotherapy or a checkpoint inhibitor, what you find is really quite remarkable, and this is what's shown here on the bottom right. So if we subset these individuals just by a quartile analysis, looking at those individuals that have maximal activation of this pathway after institution of therapy, what we find is that these individuals have more than a 50% increase in actual response rate relative to those individuals that have a minimum increase in this pathway or less or more subtle activation of this pathway after immunotherapy. And this is really, really critical. Now, this approach and this framework, as you can imagine, can also be applied within the context of cellular therapies for being able to identify those individuals who are most likely to respond. If we think about a framework of using these dynamic biomarkers, specifically within the context of drug-induced toxicity, again, whether it be for cell therapy with cytokine activation syndrome or in the setting of a gene therapy, the way we typically approach this is taking these individuals and these samples, again, whether they be preclinical models or humans in which they've been exposed to a particular therapy, assaying their blood at various time points, analyzing those blood on our RLCMS systems, again, measuring 15 to 20,000 small molecule biomarkers, and then using statistics and data to identify biomarkers of early toxicity. Now, this is where the database can be absolutely essential, in which we can take these biomarkers now and we can validate them in our database, asking essentially, in this instance, if we're seeing effects on myocarditis in the setting of AV vectors, which is a devastating serious adverse event, we can ask, how do these same markers map onto the development of cardiac toxicities in the setting of myocardial infarction or heart failure or other cardiotoxicities over time? And this way we can begin to validate very quickly a marker of drug-induced toxicity across more than 100,000 diverse individuals with related phenotypes of interest. Similarly, within the cell therapy space, we use these small molecule biomarkers within various stages of the development. One, to optimize patient characterization, both for those individuals that are cell donors, as well as patients who may be receiving these therapies, in a similar way to HLA matching, to understand those individuals who are most likely to provide healthy cells, as well as those individuals who are likely to have minimum cytokine release syndrome in the setting of exposure to these various therapeutics, as well as understanding responder, non-responder status. In vitro and cellular systems, we can use these biomarkers to evaluate cellular handling, preparation, expansion, and to really characterize these products that we know can be extremely heterogeneous. And then ultimately, after these products are introduced into patients, particularly in the setting of oncologic therapy, to be able to monitor these individuals, to develop circulating markers that read out cell durability and performance, as well as tumor killing, once again, in a means to expand our underlying therapeutic index. Now, this framework is really around aligning the right patient with their specific disease and the right therapy. And as you can imagine, this framework, while generic, is particularly important in the setting of cell and gene therapy, in which particular adverse events can be absolutely devastating. When we think about biomarker applications within the context of identifying the right patient, it's really about identifying responders and non-responders, patient stratification and clinical trial enrichment, safety profiling, once again, identifying those individuals who are likely to have an adverse event or not an adverse event, and companion diagnostics. Similarly, when we think about characterizing disease states using these biomarkers, it's really centered on target identification and validation, understanding disease mechanisms and early disease detection, as well as disease progression. And ultimately, in the setting of cell and gene therapy, it's about aligning the right therapy with the right individual, understanding dosing strategies, product characterization, particularly in the setting of cellular therapies that can be quite heterogeneous, understanding target engagement and silencing or activation, particularly in the setting of gene therapies, and ultimately understanding toxicology and adverse events in a way that, again, allows us to identify these individuals at the earliest time points, to treat them appropriately, and to really advance these truly transformative mechanistic studies. Now, I just want to take one final moment and to provide an overview of Sapient. We really feel that we're incredibly fortunate to be working with a number of individuals who are really at the forefront of cell and gene therapy. And I should say that we serve only as a purpose-built CRO, so we do not develop drugs ourselves, but rather we provide state-of-the-art bespoke small molecule biomarker analysis, what again would traditionally be called metabolomics and lipidomics, both with data generation as well as statistical analysis, as I just reviewed. We're really centered on high throughput mass spectrometry technologies, and these technologies allow us to have very fast turnaround time and measure thousands of markers in biosamples, whether they be in vitro biosamples of a cell therapy, preclinical models of a gene therapy, or ultimately patient samples all the way from a phase 1 through phase 3 trial. We enable cross-validation of discoveries using our longitudinal in-house human biology database that includes both paired phenotypic measures, cytokine and proteomic measures, inflammatory measures across hundreds of thousands of individuals. We operate as a CLIA-certified, CAP-accredited clinical laboratory, and this allows us to very rapidly translate these discoveries and to implement these clinical discoveries at the earliest time points in order to advance forward a clinical program. We provide very simplified per sample pricing that includes not only data generation, but also biostatistical analysis, and believe that our goal is to turn around this data to you as quickly as possible so you can advance forward your important drug discovery operations. And so typically we have a turnaround time of less than 60 days. And fundamentally, we believe the best data comes through close collaboration. We view ourselves as simply an extension of our sponsors' laboratories, and this allows us to really optimize around customization around your specific question of interest with full data transparency and transfer of all raw data back to our sponsors. I'll go ahead and stop there. I'd be happy to take any questions. I just want to say that this is our website, www.sapient.bio. We would welcome anyone who may be interested in chatting further and double clicking into any of these areas, whether it be the technology, the data assets, or how you can leverage these approaches within the context of your cell and gene therapy program. Here's our phone number. Our email is discover@sapient.bio, and certainly anyone can use the QR code here to schedule a meeting. And I'd be happy to take some questions time allotting.
Great, thank you so much, Mo. That was a great presentation, and we do have some questions, and we do have some time for additional questions. So if folks have more questions, go ahead and enter them into the questions box. But let's get started here with the first one. Can you please discuss the translation of these biomarkers and how you are able to apply these markers within the context of clinical trials?
Yeah, absolutely. It's a great question. And so again, this is not discovery for the sake of discovery, but rather the real fundamental goal here is how do we make discoveries as quickly as possible that can be rapidly translated back to clinical process, whether it be as part of a clinical trial implementation strategy or fundamentally in the care of patients. Now, these technologies that we discussed really represent just the first stage in a multi-stage pipeline. We use these technologies to measure thousands of markers in a subset of samples, typically are using statistical analysis to identify the key markers, validating them both technically using orthogonal systems as well as biologically in our database. And then because we have a high complexity CLIA CAP accredited laboratory, we can translate those discoveries very quickly, developing very simple ELISA-based or triple quad mass spec-based assays that measure those key biomarkers. And we can then translate those assays back to our sponsors, to another CRO, or operationalize them internally under CAP and CLIA as part of a clinical trial. So all of that happens simultaneously as part of the discovery process, and this is what allows us to go very quickly from initial sample ingestion and R&D, identification of a marker, and then ultimately translation.
Thank you. How do small molecular biomarkers of target engagement compare to traditional genetic or protein biomarkers?
Yeah, it's a really, really good question. And it's essential to understand that this is just not another data modality and that there's nuance with each type of data that we're measuring in people. Now, we provide proteomic services as well as cytokine and other measurements, as I mentioned earlier. And there's some real physical chemical differences that are inherent to each of these modalities. Now, when we think about small molecule biomarkers, what makes them unique is, once again, they're produced intracellularly in every single cell within our body, whether it be a healthy cell, a disease cell, a cell that's exogenous, that's introduced as part of a cell therapy, a microenvironment cell, et cetera. But the goal of the cell is to make this intracellularly and then to extrude it into the extracellular space and ultimately the blood. This is why when we're assaying these thousands of markers in blood, we're having deep insight and a glimpse into what's happening in the localized tissues. Now, this is a little different than protein measures or large molecule biomarkers or genetic markers, which, as you're well aware, are produced intracellularly in great abundance, but these markers tend to stay intracellularly. It's energetically extremely unfavorable to take a protein and to spit it out into blood, which is why only a very small fraction of the underlying proteome ends up in the blood. And with genetic materials, even less, as you can imagine, unless there's a substantial cellular rupture. And so in a way, these molecules from a practical perspective are providing a view or a telescopic view into what's happening in the tissues in a way that tends to be far greater than what we traditionally see with proteomic or genetic measures.
Thank you. You touched on integrative analyses. Can you speak more to the benefits of combining metabolomics measures with proteins and genomic biomarkers?
Yeah, absolutely. I'd be happy to. And that, as you're correctly stating, there's no one magic bullet. We firmly believe that small molecule biomarkers provide some of the greatest amount of information. But as you can imagine, layering these small molecule biomarkers onto other data assets, whether it be genomic measures, whether it be markers of mutational status, whether it be traditional proteomic measures or cytokine measurements, really is where the power increases for true discovery. Now, there's a variety of ways in which we can do multidimensional integration. It's fundamentally based upon the question at hand. For instance, we can do sort of very complex feature analysis where we're looking at proteins and cytokines together with these metabolites to identify individuals who are at greatest risk for a cytokine release syndrome in the setting of a cell therapy. Alternatively, we can do integrative analysis between genomic measures and protein measures and these small molecule metabolites to begin to understand the origins and for Mendelian randomization, as well as to ensure effective target knockdown of a particular target in the setting of a gene therapy. Similarly, we can use these cytokine measurements to understand small molecule biomarkers that may associate with particular inflammatory states that may be critical to both cell and gene therapies. And so there's many ways in which this data can be integrated with one another. And again, it's very much dependent upon the underlying biological question, but being able to do that integration, which is what we have deep expertise in, is what allows us to really leverage these processes for true discovery.
Great. I think we have time for one or two more, if that's okay with you.
Please.
Regarding toxicity, there are already a number of markers like NABs, complement factors, et cetera, that predict drug-induced toxicities. How are small molecule biomarkers different?
Yeah, it's a great question, particularly in the setting of gene therapies. You can imagine there's a number of assays around complement factors, around NABs, around drug-related antibodies and prior infections. And those are great on a gross scale for identifying a subset of individuals that are likely to have an adverse event in the setting of an AAV vector, for instance. Now, the challenge is the majority of patients that still have severe toxicity in the setting of a gene therapy or in cell therapy are not captured by these traditional metrics. And so there really is a fundamental need to have new biomarkers that allow us to bridge that final gap and identify those 30% of patients that are not being omitted from these clinical trials, but ultimately develop a severe adverse event in the setting of a cell or gene therapy. And so while I think these markers, again, whether they be NABs, T-cell measures, traditional cytokine measures, antibody-related drug toxicities, et cetera, while these have been absolutely essential in starting these gene therapy programs, there still is a ways we have to go to be able to identify effective markers that identify all individuals who are likely to have an adverse event. And that's where these small molecule biomarkers, as well as the proteomic and cytokine approaches that we sort of endorse, can have huge effect.
Thank you, Mo. I think we have time for one more.
Sure.
Beyond evaluating the gene therapy itself, what about the patient? Can these measures be used as diagnostics to determine patients suited for gene or cell therapy?
Yeah, absolutely. And this is a key application. While we're thinking about these tools as really a drug development, also the deployment and the implementation is where these biomarkers can have huge effect, as you're suggesting. So when we think about patient selection, there's a variety of different approaches by which these may be efficient and effective in helping. You can imagine for gene therapies where these are oftentimes centered in rare diseases, developing circulating biomarkers that can be used for diagnostics for identifying particular patients who have a given disease can be extremely important, particularly for diseases which are marked by complex genetic milieu in which there's not a single gene mutation, but there can be dozens or even hundreds of potential gene mutations. And we've had great success in identifying circulating markers that become diagnostic tests. Similarly, if we think about these from companion diagnostics perspectives, as we know, there's typically a portion of individuals that respond to a particular gene or cell therapy, and being able to identify baseline markers that substratify responders versus non-responders can be extremely important in identifying the right patients in which to go into. Now, while we talked typically about blood and urine and tumor and tissues, we can also apply these within the context of interesting other biological specimens, whether it be aqueous and vitreous humor fluid from the eye, or even tears as a mean for identifying thousands of markers that may read out particular gene therapies in localized sites, including in the eye.
Well, Dr. Mo Jain, thank you so much for a wonderful presentation, and thank you to Sapient for partnering with us as always. And this presentation has been recorded and will be available on demand. Again, thank you so much and have a great day. You may now disconnect from the webinar. Have a great day.